I'm Jonathan Carter with the Autism Science Foundation, and we're here with Rhonda Charles. Rhonda is a PhD student in the Department of Genetics and Genomic Studies at Mount Sinai School of Medicine. She's also a 2010 ASF Grantee. Her grant was entitled The Humanized AVPR1A Mouse, a preclinical model for determining the role of AVPR1A in autism spectrum disorders. So let's jump right into it. Can you please explain the role of the AVPR1A gene and how it affects autism?
Okay, so AVPR1A is one of the three major receptor subtypes that interact with arginine vasopressin. And it's very important throughout the whole human system, ranging from peripheral importance in the kidneys, vasculature, and liver for a lot of very important regulatory functions. So what we're really concerned about is the role that it plays in the brain. It was first studied a lot in lots of rodents, especially vole studies, and what they have found is that AVPR1A is actually really important in modulating behaviors that are related to autism spectrum disorders, such as social behavior as well as anxiety. So later studies that were performed with human subjects looked at both clinical and non-clinical subjects. With the clinical subjects, which would be autism patients, they found that there was an association between autism spectrum disorders and some of the polymorphic sites in the AVPR1A gene. In non-clinical studies with, typical people, they found that the gene was actually really important for dance and musical memory, as well as some social behaviors such as altruism and pair bonding with humans. So altogether, a lot of the rodent studies as well as some of the human studies have indicated that AVPR1A is really important in modulating behaviors and social aspects that obviously are very relevant to autism spectrum patients.
Can you speak to some of the smaller areas that you focused on in your research?
Right. So my research focuses on a mouse model that we've created. It's a humanized mouse model. So essentially what that means is that we've inserted the human form of the AVPR1A gene into the mouse system. And why we did that is because previous studies have shown that upstream areas of the gene are thought to be very responsible for controlling behavior by controlling expression pattern throughout the brain of that particular gene. So we theorized that by creating this humanized model, we'll be able to have a more relevant in vivo system where you can kind of bridge the rodent studies to the human studies and create this kind of, in-between system where you can look at the role of human AVPR1A, you can look at what regulates its expression, and also you can think of it as a preclinical model for testing a lot of therapeutics that are geared towards the system.
So in that vein, down the road, why do you think this research is important to families and individuals affected by autism?
So whenever you create a mouse model, the ultimate goal is always for it to have some sort of relevance, and translation to the human system. So when we created this mouse model, it was more of an investigation of how is this gene important and what could be possible targets towards treating the autism spectrum disorders through this gene. That's why I think it's really important first step, as a kind of first step for screening the biochemistry of the gene, and thereby you can have a better understanding of what therapeutics will be able to target this system, target the pathway, and target the pathogenesis of autism spectrum disorders. What
are your next steps? What are your plans for your future autism research?
So right now I'm still finishing up my dissertation work, but we have a nice new idea about investigating, the regulatory elements that control AVPR1A expression. But then when you think of the larger picture in terms of what I want to do in life, I definitely want to stay within the field. Being involved in this kind of research and having the connection through SIVA Center with actual clinical studies, my basic science has been able to blossom and I've been able to think beyond the bench. So I definitely am interested in staying in the field, but probably wanting to branch out more in terms of the direct connection with patients and with, people affected with psychiatric disorders as such.
How has being an ASF grantee helped you?
Okay. So besides the financial support that I've been provided, I get a lot of academic support as well. I was able to attend IMFA in 2010 with the money allocated from the grant. And at that event, I was able to meet lots of colleagues in the field who not only study the basic science, but also study clinical aspects of the autism spectrum disorder. And, you know, it was sort of a way to put a face to a disease that I just know in the wet lab system. So the grant setup is really important for allowing young scientists like myself to really blossom in a way that exposes us to many different aspects of the research going on, and to understand that this is, you know, more than just a mouse on a table and I'm classifying it. It's about really working towards a goal of treating patients in the future.
Well, Rhonda, thank you so much for being here.
No problem.